right? Hypoxia-inducible factor 1 alpha is a protein that in low oxygen conditions goes into your nucleus and turns on transcription of genes and turns off transcription of genes that help you respond to low oxygen. Isn't this cool though? When oxygen's around, oxygen acts to change the structure of HIF1 alpha so that it's destroyed by the proteasome. The protein HIF1 alpha is degraded in the presence of oxygen. So this protein is only stable and around when there's no oxygen. So it goes and does its job, turning on transcription. One of those cool things that just happens. So when oxygen is present, there's no HIF1 alpha. Absent, you get changes in transcription. So the question of this paper that we're going to look at next week then is, does hypoxia have an effect also on translation? We know that through HIF1 alpha, low oxygen concentration does something for us. It changes gene transcription through this transcription factor, HIF1 alpha. So what happens in translation? Okay, so go through pretty quickly the basics of translation, and then there are a few things that you'll need to know specifically for next week. What is a polysome? How do you detect them? Talk about 4 different approaches. We've already talked briefly about RNAi, and then 3 specific things that they do in this paper that we haven't talked about yet. PARCLIP, love the acronyms. RNA immunoprecipitations, and using actinomycin D, which is one of the treatments that you'll see in the paper. And then I'll end with a model or the author's thoughts of how this all works. So you'll get I'm telling you what you're going to hear next week. The punchline. So presumably there are 4 steps to translation, if you believe this figure. So initiation. What do we need on messenger RNA to do the translation? What's the end game? What's the organelle or the structure that actually does the job of translation? That thing. Yep, the ribosome. So the goal of translation initiation is to assemble a ribosome on a messenger RNA molecule. Right? Then the ribosome can read the RNA code and produce protein, chain of amino acids. The first step, or one of the first steps, is the binding of the 7-methylguanine cap that's put on the 5' end of messenger RNA. If that doesn't mean anything to you, that's okay. But everything starts at the 5' end of the messenger RNA. And there's a protein, 4E, which is technically EIF4E, eukaryotic initiation factor 4E, that binds that cap, and it recruits other proteins, and those other proteins, through these many connections, recruit the small subunit, the 40S subunit of the ribosome. And then the rest of the ribosome builds, so 4E binding the cap is the first step towards translation initiation. It recognizes, hey, that's a messenger RNA, it needs to be translated. Hey, come on, you other proteins, get over here. Let's build a ribosome. Then the ribosome scans along the messenger RNA, finds the initiation codon, AUG, starts translating, stops translating in termination, falls apart, the ribosome does, and then you've made a protein. Nah. There's differences between eukaryotic and prokaryotic ribosomes. The subunits are different sizes physically, so we call them different numbers, but it's basically the same thing. Okay. And yeah, that's what happens inside the ribosome. So take a little bit closer look at initiation, elongation, and termination. So I've already said this part, that EIF4E binds that cap and brings all the other proteins. Then you get assembly of the ribosome. Wow, I've got a lot of good for me. Okay. And this again is meant to cartoon that assembly process, where EIF4E binding is the first step. It interacts with EIF4G, which interacts with another protein, which brings that 40S subunit in. It starts building the ribosome. I did not know that this actually happened, this looping. Why is that beneficial? So EIF4G also interacts with the poly A binding protein that binds the 3' tail of the messenger RNA. So the cap is right next to the poly A tail. What direction are these ribosomes moving? Clockwise or counterclockwise?
Clockwise.
They're starting at the cap and going, so the cap's the 5' end, and then they move that way as they translate towards the 3' end of the RNA. So what happens when this ribosome is done translating? What happens to the ribosome? When it hits a stop codon, it falls off. And then if you're trying to make lots of this protein, what do you have to do next? What would be the next step? Make another ribosome. Get it going again. Assemble a ribosome, make it translate. Assemble a ribosome, make it translate. Make lots of protein. So this looping presumably is important, as it says here, so that ribosome subunits are recycled. That is, when they fall off, the large and small subunits, they're right there next to the 5' end again. So that they get immediately recruited by EIF4E and the other eukaryotic initiation factors, get right back on the messenger RNA and go again. It's like going on a ride at Disneyland. You do the ride, you get off, you run to the line again, get in line, do the ride again. Okay. So hopefully, at least, if nothing else, I've impressed upon you the EIF4E is critical for translation. It's the first step, and it helps recruit ribosomes. Then, in the introduction to this paper next week, you'll read that EIF4E doesn't exist under hypoxic conditions. When there's low oxygen, well, when there's normal oxygen, this protein mTOR adds phosphor, it's a kinase, it adds phosphate atoms to this protein called the 4E binding protein, 4EBP. Those phosphate molecules keep it off of EIF4E. When you move into a situation where there's low oxygen, as is shown over here, I was going to write it over here, there's no more mTOR, it gets degraded. So 4EBP is not phosphorylated, and it grabs onto eukaryotic initiation factor 4E and prevents it from binding 5' caps of RNAs. So normally, just using EIF4E, when you move into low oxygen, you have no more translation because there's nothing to bind that 5' cap and start translation. And then the authors point out, well, this doesn't make any sense because we know some translation occurs during hypoxia, low oxygen. So the core question they're asking is, if EIF4E is not available, how does translation happen? How does the ribosome get recruited and assembled? So that's all initiation. Here's elongation again, just the basics. The ribosomes read 5' to 3' and add amino acids that come in on transfer RNAs, and you synthesize the amino acids or the proteins for the C terminus. When that ribosome hits the stop codon, then transfer RNA, there is no transfer RNA, or it doesn't transfer amino acids in the stop codon, that causes the ribosome to dissociate, and you've got a full-length protein. So here's what a polysome is, and there's polysome profiling in this paper. So this is like riding the ride at Disneyland. Lots of people are on the ride at once. In this case, it's not just one ribosome per messenger RNA per unit time, I guess. You can have lots of ribosomes at the same time translating one messenger RNA, and that's what this shows, including a cool electron micrograph. So the authors will be measuring whether or not certain transcripts are polysomes or not. What does this tell you about a transcript? If it's a polysome, if a messenger RNA has lots of ribosomes on it, what does that tell us? It's the obvious answer. Yeah, it's being translated heavily. Yeah, so they're going to use this as a metric to ask, are certain transcripts being translated under certain conditions? So if they're trying to figure out how hypoxia is permissive for translation, then you have to be able to show that translation is occurring. And one way is to ask, are messenger RNAs part of polysome? So this is kind of how polysome profiling works. If there's a messenger RNA that's associated with a lot of ribosomes, it'll be denser. So if you centrifuge it in a sucrose gradient, it goes farther because it's heavier than something that doesn't have any ribosomes on it, which is less dense. So you just load a cell lysate into a column, centrifuge it for a while, and then out of this you take fractions. You collect the different fractions, and you get a profile like this, where you've got essentially something equivalent to protein content on the y-axis and fraction number on the x-axis. So we know that the individual subunits, the 40s, 60s, and 80s subunits are pretty small, so they're on the left side. They're the early fractions, the light fractions. But then you start seeing this really cool oscillation. Those are polysomes. So you can say, for example, although I'm not an expert at reading these, that's 1 ribosome, 2 ribosomes, 3 ribosomes, 4 ribosomes, 5 ribosomes, etc. So if you have a standard, to compare this to, you know exactly if you're in which of those peaks corresponds to how many ribosomes are loaded onto one messenger RNA, with more ribosomes being to the right. So this is the way they can ask, is a messenger RNA being translated? Is it part of a set of polysomes or is it not? And they'll be using a lot of RNA, which we talked about previously. I just wanted to give a quick reminder that if you have double-stranded RNA, it's a single molecule of RNA, but it folds back on itself to make a hairpin structure like shown there. That can be exported out of the nucleus and gets processed by that protein dicer that turns it into what's really a double-stranded piece of RNA, not just held together, not a single strand, it's two strands now. That hairpin's been cut off. And that single-stranded RNA, after it's separated, gets incorporated into the RISC complex. And then that protein RNA complex goes around and tries to find messenger RNAs that are complementary to that short inhibitory RNA, the siRNA. And if it finds an RNA that matches a short RNA, then that RNA gets degraded, this one. Gets cut up into little bits, so it can't be translated. So this is one technique the authors will use to knock down expression of certain genes. You get rid of gene expression by basically providing cells with double-stranded RNA that's targeted to specific genes. What's RNA IP? What does it sound like? There's an immunoprecipitation of RNA. You're right. Yes, good. Excellent. So you can't immunoprecipitate RNA. I'm sorry, you can't. Because antibodies detect protein epitopes. RNAs are not proteins, so RNAs don't have shapes that antibodies detect. As far as I know. Somebody prove me wrong. If there is an RNA that an antibody can detect, please tell me about it. So the goal is, right, if we're studying translation, they're trying to find what? If there's no EIF for E, this thing that binds the 5' cap, what are they trying to find in this paper, do you think? What's a hypothesis for how translation can work if you don't have the main thing that recruits ribosomes? Yeah. There's another protein that does it. It's just not EIF4E, it's EIF something new that no one's ever seen before. So if you want to do something like that, if you want to find a protein that binds RNA, then you might do an RNA IP. It's actually the opposite question. It's if you find the protein and you want to know if it's binding to RNA, and if it's binding to specific sequences of RNA, then you can use this approach. So say you've identified your suspect protein, the substitute for EIF4E, and you want to know what sort of messenger RNAs is it binding. How do you get the identity of the messenger RNA that's bound to your protein? That's how you do it. You immunoprecipitate the protein, and then you just collect whatever RNA comes with it. Sound easy? It's conceptually pretty straightforward. It's an immunoprecipitation. You have an antibody that binds your favorite protein that you're interested in studying, and as long as that protein stays bound to the RNA it was originally bound to, then you just collect all the RNA and use it for, as it shows down in the bottom there, quantitative RT-PCR, reverse transcriptase. So you turn the RNA back into DNA, use it for PCR, or you sequence it. So you find the molecular identity. What are the messenger RNAs? What are the genes, basically, that protein is binding? Of course, the critical point in what I just said was, assuming that the protein stays bound to the RNA it was interacting with when you pulled it down. So the really conceptually or experimentally hard thing to do is to keep the conditions in the IP ideal for keeping proteins interacting with their RNA while you're messing with them by immunoprecipitating. But people have worked out, it's hard, but it's doable. So people do this. So you pull down these proteins and then collect the RNA and study the RNA.